Good evening. I'm Fred Allendorf from Biology. It's my honor tonight to introduce our presidential lecture speaker, Steve O'Brien. And I'd like to thank President Engstrom for supporting this, and also the Montana Ecological Infectious Disease Program, which has also sponsored Steve's visit. We're really all lucky tonight to have Steve here. Steve is truly an amazing person, an amazing scientist. He's had about 3 or 4 different careers in the time that he's been working in this area. So tonight he's going to be talking about HIV and AIDS and his work at NIH. But a lot of us know Steve more for his work in conservation genetics, as well as his work for the National Cancer Institute on HIV. And Steve has been a leader in applying the principles of genetics to issues in conservation. And this is one of Steve's favorite species. Steve has specialized in work on cats, and this is the Nepal Tiger Genome Project, which has come out of a lot of Steve's work. So Steve was head of a lab at NIH for 25 years, was the editor of the Journal of Heredity, which is the journal of the oldest genetic society in America, and he fulfilled that role for 20 years. I've known Steve for about 30 years. He got his PhD in Cornell in 1971, working on Drosophila, the little fruit fly, as a population geneticist. And I guess what I really enjoy the most about Steve is that he personifies the importance of understanding evolution for trying to do any work in biology. So Steve is going to be going to Russia. He just retired from NIH. And he's received a mega grant to start an institute in Russia on genomics, comparative genomics. And the institute is going to be named after Theodosius Dobzhansky, who is a Russian who spent most of his career here in the United States. And probably most of the people in this audience know Dobzhansky from this quote, famous quote that we often hear, that nothing in biology makes sense except in the light of evolution. And so I'd really like to welcome Steve tonight to express his career as an evolutionary biologist, applying it to issues in HIV and human medicine. So Steve.
Good evening, and thank you very much for coming. It's a special pleasure to come to the University of Montana here in 2012. I think the last time I was here was in the late Pleistocene, when they were using 2x2 slides, as Fred reminded me of earlier today. But I'm happy to still be standing and to be able to tell you a perspective that only an old-timer who has worked in a horrific epidemic could tell you. And that is the story of 3 decades of genomics and AIDS tiptoe towards a solution. This is a photograph that was taken in 1996 on Columbus Day from the top of the Washington Monument. It is the AIDS quilt. It has approximately 40,000 emblems in memory of a patient or a couple who had died of HIV at that time. This epidemic began early in the late 70s, when a couple of unusual infections appeared in actively gay men in San Francisco, Los Angeles, and New York. It rapidly spread throughout America, throughout Europe, throughout the world, and in a very short period of time took an untold number of lives. The cumulative HIV infections as of today is about 65 million, and there have been about 30 million people that have died of this disease, a disease for which there is no effective vaccine, for which we have treatment which is not a cure, but which really just stars off the virus for a long period of time. But suspending the treatment nearly always, in fact, it always results in the virus rebounding back from latent reservoirs that sit throughout the body. There are approximately 2.6 to 3 million infections every year, and it has become the most deadly epidemic since the Black Death back in the 15th century. It is a disease which my children have never lived a day not thinking, that is, they've never lived in a time without AIDS, and they were actually with me walking among these emblems back in 1996 when we listened to Elizabeth Taylor talking about the tragedy that we needed to get to. Well, when this epidemic began, I thought at the time we were working on things like cheetahs and pandas at the National Cancer Institute, and I decided that I needed to do something a little more closely related to human health so that I didn't get fired from my new job as the director of this laboratory. So I embraced the help of my friends who were working in human genetics and in HIV surveillance and asked them what they thought about looking for genetic variants that might explain some of the heterogeneity associated with the AIDS epidemic. Schools of public health study epidemiology, heterogeneity, and epidemics so that they can help understand better the specific causes and influences of disease progression in a population. I began looking for AIDS restriction genes by enlisting the help of these epidemiologists and telling them to do the following: continue to gather data about individual patients, but send me a blood sample. I will turn it into a lymphoblastoid cell line by a transformation procedure with a virus called EBV, the same thing that causes mononucleosis, and then I will have an immortal store of DNA from that patient. We wanted to discover human genes with polymorphic variants that influence the outcome of HIV exposure. Why do some people get infected and others do not? Or infection. Why do some people get AIDS quickly while others postpone it for up to 10, 20, even 30 years? We wanted to discover these genes to discover natural solutions that would inform diagnostics, prevention, and help maybe even developing vaccine, but further to identify new avenues of natural for therapy. We started with epidemiologists who were developing what are called AIDS cohorts, which are populations of people exposed to HIV due to their high-risk behavior or because they had received contaminated blood products in the early 1980s before the discovery of HIV, and this was between 1978 and 1983. These included hemophiliacs, as well as recipients of blood transfusions and IV drug users who were sharing needles in city slums when the incidence of HIV infection was something on the order of 80%. We gathered, you can see from the enrollment date, that there were most of these cohorts began many years ago in the early 1980s and have been followed ever since. So we had a longitudinal study, and there were thousands of patients collected, and today we have over 10,000 patients. And these are the people who we use to look for HIV restriction genes. Now, to keep our study a little bit focused, we concentrated on specific stages of HIV infection. First, we asked, are there genes that can influence whether or not somebody gets infected? Some people get infected, a lot of people don't. Well, actually, most people do become infected when exposed, but some people don't. Is there a gene that influences the rate of progression? Before the powerful drug therapy called HART, or highly anti-retroviral therapy, before 1996. Then we looked for genes that influence the kind of AIDS-defining condition people get. People don't die of HIV infection, they die of a lymphoma, or they die of a Kaposi sarcoma, or they die of a wasting disease, or some other opportunistic infection, which normally the immune system dispatches pretty well. We looked at the viral load, which is the concentration of the virus that's present in patients over a period of time. We looked at the number of CD4-bearing cells, the indicator cells that HIV actually destroys, which when they fall below 200 is an indicator that the person is going to die of AIDS. We also looked after 1996, when the powerful antiretroviral drugs were developed, what the heterogeneity was in that epidemic. Are there genes that influence whether or not it works, whether or not there are adverse events? And this search for genetics was the beginning of using the human genetic technologies to understand genes that influence complex diseases. In 1996, a group of workers in New York and Philadelphia described the process by which HIV enters cells. They bind to a molecule on the surface called CCR5, indicated in this slide. And then this is a requisite for HIV to enter cells. So we immediately wondered whether there were any mutations that were common in this gene. So we sequenced a number of people for the CCR5 gene and discovered that many of them carried not a single base mutation, but a whopping chunk of the gene was removed, 32 base pairs, which altered the codon usage. That is to say, it altered the, it altered the reading frame of the gene and led to a truncated protein which put the wrong amino acid in. In people that inherited 2 copies of this gene, 1 from each parent, the truncated protein would go to the endoplasmic reticulum, which is a sort of a filtering part of the cell, that recognized this as a bad protein and it would simply destroy it before it made it to the surface of the cell. This meant that people with 2 copies of CCR5 delta 32, the mutation, actually could not put any CCR5 on the surface of their cells. So when we went to our cohort, and we had, as I said, over 10,000 of them, this is the frequency of individuals that had the 3 different possible CCR5 genotypes among individuals that were HIV negative versus HIV or positive. If you had 2 normal copies, the frequency was the same in negative or positive HIV people. If you were heterozygous, 1 of each copy, it was the same. But there was an unusual thing here, which is that individuals that were homozygous for delta 32 were never found among HIV positive people. The reason was HIV couldn't get into the cell. They had a shield. They had a protection. There was no doorway. No matter how they behaved or how often they had been exposed to HIV, they would never become infected. You can't imagine how many people went out and got their genotypes done, and then they would come up to me and say, "I'm homozygous for delta 32. Isn't that cool?" I said, "Well, I don't know. We'll talk a little more." But the truth is, it was a very important discovery. Now, individuals who are heterozygous have 1 copy of the normal and the other. They do become infected. Is there a pointer that I could use here? Someplace? A laser pointer? Huh? You don't have one? Okay, sorry. Okay, basically what happens is that people who become infected with HIV, as is shown here, do progress to AIDS, but if they're heterozygous for HIV, they actually delay the onset of AIDS slightly, about 2 to 3 years. This is a survival curve. It simply shows on the top or on the bottom the rate of progression to AIDS. In about 8 years or so, people who have 2 copies of the normal gene get AIDS, about half of them do. But if they have 1 copy, they slow it down 2 or 3 years. So it was basically what geneticists call a dominant restriction against progression and a recessive restriction against infection. The reason for the slowdown in progression to AIDS is because individuals who are heterozygous actually develop lymphomas less frequently. And that's shown by the frequency of delta 32 in heterozygotes among positive versus negatives. There's no difference in the Kaposi's patients or the opportunistic infections or the pneumocystis carinii pneumoniae. But the lymphomas, the individuals who are positive for delta 32, that is, have the protection or partial protection, they have half the rate of lymphoma. This was the first AIDS restriction gene. There were several others that were discovered after it. One of them is the major histocompatibility complex. In people, it's called HLA. It's a group of 220 genes that are involved in recognition of infectious agents. It's extremely variable, and it's variable because the genes have a repertoire to recognize peptides that come in from invading viruses. So the more diversity that you have, the better off you are for recognizing viruses that you've seen before, or even viruses that you haven't, or bacteria. Well, when we sampled people who had different genotypic dispositions for HLA, for the class 1 genes, the ones that present peptides to the what's called the cytotoxic T cell system, which recognizes viruses, we discovered there was a difference. People that were heterozygous at all 3 class 1 genes progressed to AIDS at the normal rate, like the blue line here. But if they were homozygous at 1, they actually progressed more rapidly. If they were homozygous at all 3, the rate of progression to AIDS was twice as fast. They basically got AIDS in half the time from individuals that did not. An interesting phenomenon, but not one that is translated quickly into any therapy. In addition, there was a number of specific forms of HLA. HLA is the most variable complex locus in the human genome. There's 500 different defined, actually there's closer to 1,000 different defined alleles, which are types. And a few of them were shown to be good for you and bad for you. One that was bad for you is one called B35. Individuals that have 1 copy of this progress to AIDS more rapidly, as the pink line shows, and individuals with 2 copies of B35, they get AIDS, all of them, within 5 years. None of them survive beyond 5 years. This is not the good genotype to have if you're basically having this. Now, B35 is actually a group of many different molecular forms. It was defined by an immunological test, but the sequences of all the different forms that produce this B35 immunology is about a dozen. And those different ones are discriminated from each other based upon the peptides that they recognize. Now, when you look down on an HLA molecule on the surface of a cell, what you see looks like a bear trap. And since this is the home of the grizzlies, you all know what a bear trap looks like. It basically has a alpha helix, which is that helix going around each side. Those are the jaws of the bear trap. And at the bottom is a pleated sheet, and inside is the peptide that it recognizes. And there's a signature for every HLA form or allele that has to do with what the residue, amino acid residue, is in the virus peptide. And in HLA B35, there is a position number 2 and 9 are the things that determine specificity. And there are 2 kinds of B35s. One has proline in position number 2 and tyrosine in position number 9. And the second has proline in position number 2 and anything but tyrosine in position number 9. We call the first one PY, the second one PX. Why am I telling you this? Well, because it turned out that the B35 effect was specific to one of these, as is shown in this slide here. If you look just, for example, at Europeans or Caucasians, the signal is entirely coming from the red line, which is a B35PX. The B35PY is identical to the wild type. So this is the signal, this is the other 2. And if you go to African Americans, a different population entirely, you see exactly the same thing, but it's not the exact same alleles or forms. It's just the ones that recognize it. So this connected peptide recognition, not just the HLA, to the effect of being highly sensitive. And the people that we had that got AIDS in 1 or 2 years all were homozygous for B35PX. This was not a good genotype to have if you get infected with HIV. Slowly but surely, we've been able to parse out the different components of risk associated with HLA molecules. This slide shows you that if you're B57, that's a good thing to have if you're infected with HIV. B27 is also good. B35 is bad, and everybody else are in the middle. They're sort of like the index. And this kind of stepwise thing has led us to realize that HLA does play a role. Now, one thing that you should understand is that HLA defense against infectious disease has been around a long time. Viruses that have survived as long as things like HIV and the deadly viruses we have today are much smarter than we give them credit for. The first thing most viruses do when they get into a living system is they figure out a way to interfere with the production of HLA because they don't want it to kill them off. So they evolved mechanisms to downregulate HLA. Nearly all viruses do it, including HLA. So what did the host do? Well, several primates in our history have developed a system called natural killer cells. And these natural killer cells are like a security detail that go throughout the body looking for cells that don't have HLA on the surface. Why? Because if it don't have HLA on the surface, they're probably infected with something that turned it off. So that whole system is a group of genes on chromosome 19, HLA is on chromosome 6, that was evolved in defense of these viruses. And the way they work is it's called KIR systems, or natural killer cells, and they look around for cells with HLA. If they have HLA on the surface, everybody's fine. Nothing happens, they go away to look for something else. But if they find one with no HLA on the surface, then they said, oh, this is a bad banana, we're going to kill this, and they do kill it. But the specific recognition of the HLA and KIR has to do with an interrelationship between the certain HLA forms and the KIR forms. So we wondered if a combination of HLA and KIR could be associated with age survival. And sure enough, it was. We found a combination that one or the other by itself makes no difference. But when you have a combination of the KIR 3S1 or BW4, what you find is that people that get AIDS in less than 3 years basically have a very low fraction of this, but people who resist it for 12 years or longer have 3 times as high, and the intermediates are in between. So you have a stepwise system where you have 2 genes on different chromosomes interacting to predict whether or not a person is going to survive. This kind of exercise we've been doing since 1996 when we first discovered CCR5. And our group and others have gone on to discover over 30 different genes that mediate HIV cell entry, acquired immunity, innate immunity, and HIV regulation. Wow, that's all pretty good. This is a listing of all those genes. I don't expect you to read all these, but the number of genes go from 1996 to 2009, and they're usual suspects, and they all have small, modest effects that have been replicated and we think are real. This is what we mean by a complex disease, a disease that has a virus, a disease that has genetic resistance, and different kinds of things. So basically, what we've gone to now is the genes that I've talked about so far are genes that we call candidate genes, genes that we guessed were involved because some laboratory experiment said, well, this CCR5 is important, or HLA is important, maybe there's a genetic difference. But recently, with the human genome being sequenced and the possibility of interrogating gene sequences across the genome, it is now possible to interrogate, or that is to test, not only the genes we can guess at, but the ones we can't. The Human Genome Project has told us that there's about 20,000 genes in the human genome. In your sperm and egg, there's 20,000 genes that determine and make you a human being. Furthermore, if you look and sequence through the DNA of several of you, you'll find DNA differences called single nucleotide polymorphisms, called SNPs. These are DNA differences. They come about every 1,000 or so base pairs. So there's 10 million of them that differentiate each of you from the person sitting next to you. Well, the biotechnology companies have come up with these genotyping arrays where they put on silicon chips 1 million different SNPs that can be genotyped. So you can take some person and genotype 1 million of his genetic differences in an afternoon for $250. That's the technology is capable of doing. That means that if you have a cohort of thousands of people, you can go throughout the genome and capture, if you will, any population genetic disturbance, which might be indicator that a person in one category, say HIV negative or resistant, is different from the HIV positive. When you do this for 1 million genes, 1 million SNPs rather, across the genome, what you're doing is you're taking little pieces across each gene that are basically trying to understand if the gene has some effect on the clinical characteristic of the individuals in the population. And what you do is you do a statistical test where you say, well, if you have a 60–40% difference between one form and another at this particular locus in the HIV infected, do you have the same thing in the HIV uninfected? If you do, you forget about that one. If you don't, you say, oh, that's important. But you do this computationally 1,000,000 times across all these individual patients. So we did this with our cohorts to look for what we considered the most robust character we could track, which was the rate at which individuals go on to get HIV disease or AIDS after they become infected. We had 6,000 study participants, 1,000,000 variants, which after you subtract out the ones that are not variable because they're not African American or Chinese specific, it comes out to about 700,000. Then you put it down on a single specific hypothesis, which is AIDS progression. And what's shown on the right is the AIDS progression result that we're kind of looking for. This is what happened with CCR5. Now, this slide illustrates the first snapshot of how we visualize where a region of the genome is important. What we've done is we've taken all 700,000 of the variants and we've put a dot in there for what the statistical p-value is for the difference between the allele frequencies in the fast progressors versus the slow progressors, or in this case, the statistical significance of the difference between the survival curve. And then you go to each one, and you wind up plotting the negative log of the p-value. So if the number says 3, it means the p-value of everything at that range is 10 to the minus 3, or 1 in 1,000. 10 to the minus 4 is 1 in 10,000, 10 to the minus 5. And what we're looking for are SNPs or variants that go very high, above 10 to the minus 7. Why 10 to the minus 7? Well, because when you run a bunch of tests like this, sooner or later you're going to get chance associations that are not real, that are statistical artifacts. This is the so-called curse of the multiple tests. But a genome-wide significance accounts for that by saying we're only going to accept p-values that rise above it. And as you can see, there's one gene or one region on chromosome 2. This turned out to be in the middle of a gene called, and there's a bunch of them. And the reason there's a bunch of them is because they are all close together, and the SNPs that are close to each other are not independent with respect to their segregation in populations. And the reason is that there are chunks of SNPs that travel together in a phenomenon called linkage disequilibrium. It's not important you understand it, but it is something that we use to help validate the finding, because we see multiple variants close to each other showing this signal. The reason that we saw it is that if we took the SNP that had the strongest signal, the one on the top of this slide right here, up with 10 to the minus 9, and we plotted the difference, we see that people that have one genotype progress to AIDS in 10 years like normal. People with one, which are heterozygous, slowed it down, and people with 2 copies, they never get AIDS at all. They're just completely resistant. If we parse that into the gay men who are part of our study group, or the hemophiliacs who are another, we see the same thing. That is, people with 2 copies never get AIDS, and the heterozygotes do get it, but not quite as frequently. This was a very powerful signal. It was the strongest one we had out of the 700,000 we tested. When we looked at the transcripts, which is the RNA produced by the gene, we discovered that there was a functional difference between the amount of transcript that was produced by the different genotypes. If the different genotypes were called CC, CT, and TT, you can see that quantitatively there is a functional difference. One of them is producing twice as much as the other. This then connects our association to a functional data. What is the gene? It's a gene we didn't know much about. It was discovered, it's local in epithelial cells, it's expressed in a bunch of tissues, and it was originally discovered in a nematode as a part of a developmental phenomena. It interacts with a group of proteins that are called SMAD, for Mothers Against Decapitogenic Homolog, whatever that means, and it interacts with the SMAD proteins that affect HIV. So we've discovered a gene that somebody had said had something to do with HIV, but we actually connected it in the population. In additional visualizations, when we have a bunch of signals in regions particularly that are below genome-wide significance, we'd like to come up with a way in which we can interrogate those a little better and look at it. But if you think about it, you've got 700,000 variants and then a bunch of different tests, a test for infection, a test for progression, a test for AIDS-defining condition, a test for the results of treatment. Those are only 4 tests, but in each test, you've got dominant models, recessive models, you've got different ethnicities, you've got different ways of looking at it, categorical versus these survival curves. There's 150 tests you can do. Well, 150 tests times a million SNPs is 150 million p-values. Now, even my most aggressive and careful graduate students can't look at 150 million tests at once. So we tried to come up with ways to help them do it. And these are computational tools, which I'd like to explain to you. For example, one's called array, and all it is a 2-dimensional heat plot. What does that mean? Well, what you do is down the left side, you list a bunch of variants that are next to each other. And then along the top, you have a bunch of tests. Like you have 8 tests for infection, 10 tests for progression, which is categorical, 25 tests for progression, which have different groups and different subsets. We have the gay men, the hemophiliacs, and so forth. Then there's the AIDS-defining condition. Then there's the therapy. And then you color code the p-value so that the low p-values like 0.5 and 0.1 and 0.001 are gray or yellow, but the stronger ones are brighter. And then you look for colors. So if you go to a region which has nothing to do with HIV, and this is a region of the gene which causes cystic fibrosis, and you look at all the variants in that region from our actual data, and you look at all these tests, what you see is what we call background. You see a lot of gray, you see occasional green, and then a few of the oranges. But the one up above, well, that's really colorful. That's the CCR5 region, okay? And what I circled is the CCR5 delta 32 gene itself. It has firing in the infection tests and the progression tests and the AIDS-defining condition tests, and then it has all the SNPs that are near it also firing, because this is a region that has a large, long block of linkage disequilibrium or non-random tag-alongs or hitchhikers, other SNPs that are going along with it. So what we look for then is we go across the genome is regions of background that look like this compared to colorful regions that look like this. And the colorful regions are the first clue. There's something going on there that's interesting. Now, we weren't quite satisfied with this because all this does is has the p-value. There's another statistic that we get out of every one of these tests, which is relative risk. It says, how much more likely are you to get AIDS or get infected? 3 times, 4 times, 5 times. That's the relative risk. It's also called the odds ratio. So if we could figure out a way to get that in here, it'd be even better. So we made a 3-dimensional plot of the same thing. And the 3-dimensional plot actually, instead of just being colored, it looks like a checkerboard, and what comes out of the checkerboard is a building. And the building is high when the p-value, negative log of the p-value is high, and the color of the block then indicates, the strength of the color indicates how strong the relative risk is. And the third thing is that there's red and green means good for you, so that means a relative risk of something that's protecting you from the effect, and red means bad for you, and that means something that makes you more sensitive. So if you look at the CCR5 region, the actual tallest thing here on the left is the CCR5 infection. Remember, nobody gets infected. This is a principal component analysis associated with the progression. This is the categorical progression test. This is a CD4 viral loads group of tests. This is a progression with a survival analysis. This is AIDS-defining condition, and this is treatment. There's 10,000 blocks there. We're looking at a region where there's something important going on. It's the CCR5 region. One of the other points is that if this is one signal all coming from CCR5, delta 32, then why are they different colors? Shouldn't they all be going in the same direction? And the answer is yes. That they're not is an artifact of the computer scientist, not the biology. Because the way the computer scientist did this was he just took the common lowest allele for each one of these SNPs along here, and then he said, is that protective or not? And that's okay if the lowest allele is always tracking the lowest frequency allele in the next SNP or the next one and the next one. But suppose the low frequency allele, the one that's 40%, is actually in linkage to equilibrium and tracking one that's over 50%. Well, then that's going to flip things. So that's easy. You just adjust it such that you have an index, which is the CCR5 big one, and then you just say, whatever that's tracking, that's the one we want to know, is it protective or not? And then we'll know whether or not there's one signal. Well, when we do that, what we mean by that is polarize. We basically, is a color code the adjacent allele that is LD tracking the index variant, and it isn't necessarily the minor allele frequency. When we do that to this, here's what we see. Everybody turns the same color. Bingo. That means it's one signal, and it's all this big block of LD, and it's all firing. They're non-independent tests. They're non-independent SNPs. Okay. So now we've got this thing. The sad part about it is that if we have this piece of paper, if you print it out and you look at it, is 8 by 10, comes out of a Xerox machine. Well, this is maybe, I don't know, 100 SNPs, and that is maybe 400 kilobases long. It takes a few thousand of these, about 6,000 of them, to cover the whole genome. So we're just looking at a little portion of the genome, the one we know is going on. But anyhow, the same thing if we go to regions we're interested in. This is the Par 3B region, the one I just showed you about. It shows you the array. You can see the strong dark signals. When you do the polarized 3D, you get the same thing. You get this, what looks like the Chicago skyline, and the background is the Kansas wheat fields. I mean, I'm just explaining to you how we're trying to look at 10,000 SNPs at once without driving your graduate students to drink, which of course none of them do. The PROX gene one is another gene that we discovered recently that has an interesting function that we are still trying to understand. But this is what the array looked like. Again, lots of color, firing and infection and progression, but not in age-defining condition, and then again in treatment. With the three-dimensional, it looks like this. Really does look like the Singapore skyline or the Manhattan skyline, maybe even the Dubai skyline, because this is a signal. And it takes into account what we know about genetics to try to find regions that may not quite hit the genome-wide significant, but are real nonetheless. Now, what I'd like to do is to mention to you that if I laid out those three things, it would be 10 feet or 3 feet long. But if I went across a whole chromosome, it might be 100 yards of those sorts of pictures. But if I wanted to go instead through the whole genome, well, that would be close to 1 kilometer, a little less than a mile. So I'd be walking along like this for 1 kilometer looking for all these regions. Well, I didn't want to do that. I didn't mind my graduate students doing it, but I did want to make it a little easier for them. So I started thinking, well, suppose we took these data and we put it into a routine, a computer routine, that allowed us to ride along the surface of this, over the top, in a helicopter. Sort of like the kids in the video games who are driving the machines and trying to shoot each other. So I had a postdoc whose husband developed video games. So I says, come here, I'll talk to you. I said, here's what I want you to do. I want you to write some code that will allow us to go along the top of this thing and to view it. Now I want you to remember, CCR5 was one of the biggest signals we saw. So now, what I'm going to do is I'm going to show you a little movie which shows what it's like if you, in fact, put this thing into gear, okay? let's just get this thing big. How do we do that? Bottom right, this thing? Okay, now here we go. Here we are, we're riding along the AIDS restriction gene highway. Over here on the left side are all the HIV infection tests. There's about 7 of them. This is the principal component analysis, which is a grouping of AIDS progression. This is a group of AIDS progression that have to do with categorical. This is the viral load in CD4. This is the AIDS progression tests that are survival. This is the AIDS defining condition, and this is the therapy. 10,000 you're looking now. But let's ride. Long. And as you can see, there's a lot of noise, different colors. This is not polarized. It's basically going along, and occasionally you'll see stuff that looks interesting. You're never really 100% sure what's interesting and what isn't. I mean, is that interesting? Probably not, but you can look at it carefully. And then the SNP that identifies is listed over there on the right. So we put all this data on an internet browser, put it out there. So people who have a gene, and this happens to me regularly, they call up, say, Steve, we've been studying this AIDS cohort in Switzerland, and we've got a signal on chromosome 17 at this particular position. Do you see this in your cohort? I says, I don't know, go look at my data. And they go to this thing, and they can type in the coordinates, and they can go right to the region and see what it looks like. And this is a good way for them to get a replication. They don't even have to talk to me. Some of them don't want to. But the truth is, it's a new way of sharing data without violating a problem. And the problem is that you can't take patients' clinical data and put it online. Scientists want to have data sharing. They think everybody ought to see everything that you do once you publish it. Well, that's fine if you're working with houseflies and fish, but you can't take some guy's clinical data and put it on a website. It violates HIPAA. It violates all the rules of privacy. So this is a step back. It takes the clinical data and the results of all the tests I can think of and puts it up there so anybody can look at my data on a website and then say, okay, it's basically replicated in the NCI cohort. And that's kind of interesting because nobody else has really come up with a solution to that. And you go along and you can stop anywhere and you say, well, that's pretty much of a negative area. There's not much going on. This is probably a low frequency, something that isn't really worth much. But now, here we're coming along here, and all of a sudden, there's Dubai again. That is CCR5 delta 32. That's the region. So we push the button, we do the polarization, it turns the color like I'd shown you a few minutes ago, although this thing has got a mind of its own right now. But let's just get out of here and go back to this. Okay. Please stop before I take away. Okay, there you go. We go to the CCR5 region, we polarize it, and what we see is that. And we say, yeah, that looks interesting. And then we can go ahead and print out a report that gives us the results of all these tests with the survival curves and everything like that. That really is an advance towards getting into these complex diseases, which goes beyond just taking the biggest hit, writing it up, and saying, I have no idea what it means. It takes into account the genetic background and our ability to use intuitive genetic inference to come up with regions that are important. Why would we want this? Well, because we want to go to the bedside. We'd like to do something better. One of the things we wanted to do for discovering these genes is to identify host factors suitable for therapeutic intervention. After we discovered CCR5 delta 32, several people said, well, can we use this? Can we interfere with HIV binding CCR5? It also binds another molecule called CD4. So a number of drug companies listed here on this slide have developed drugs that they have attempted and gone into clinical trials to interfere with HIV binding, a whole new area of HIV entry inhibitors. And since 1996, when delta 32 was discovered, 3 of them have become approved. That's pretty good. They're not perfect because they're expensive and they have to be used by injection, but at least they're different from the other drugs we have. this is a timeline of HIV drugs. The first one is AZT from 1997 up to 2011. And what you'll see is many of these are anti-HIV enzyme inhibitors. But the three that I'm going to circle right now, the three that I just circled are entry inhibitors stimulated by the discovery that people who are homozygous for delta 32 do not become infected, slow down the rate of AIDS, don't get lymphomas, and are pretty much protected against AIDS. That's what we want with these kinds of discoveries. Translation. Now, in the final few minutes, I want to tell you something about the CCR5 delta 32 that's kind of interesting. And that is, why in the world would there be a mutation that knocks out a perfectly good function floating around at a frequency of 10% in Europeans? What I didn't tell you is we don't find it at all in native Africans or Asians. It's just found in Europeans and European Americans, and the frequency is 10%. If the frequency of a gene is 10%, the frequency of homozygotes is about 1%. When was CCR5 delta 32 born? Didn't know that yet, but we're going to try to get at it using the tools of molecular evolution. What can we learn about AIDS resistance from the history of the variant? Well, as Fred said, one of my teachers, Dubzhansky, wrote long ago, nothing in biology makes sense in light of evolution. So we took a page out of 30 years of molecular evolution to get at this issue. A couple of pieces of data. First of all, delta 32 is present only in Europeans, but its frequency defines a gradient. It's highest around 15% in northern Scotland and up in Sweden, about 10% in Germany and France, and down 5% in Italy and Greece and Turkey, and 0% in Saudi Arabia and in Africa. Furthermore, we know from other people's studies that the European and the Asian ethnicities derive from a migration out of Africa about 120,000 years ago, where they outcompeted, mated with, destroyed the Neanderthals and the other human ethnicities around at the time, and established the Caucasian and the Asian peoples in the last 60, 70,000 years. Well, since delta 32 is only found in Caucasians, it must have occurred after that, in this position right here. Okay, so we know it's a little bit young, less than 50,000 years that it was born. We also know that it defines a gene frequency gradient. And the kind of thing that we see that forms these gene frequency gradients is some sort of breathtaking selective pressure advantage associated with delta 32 that caused it to rise up in the north, but then gradually go away. Short term, didn't occur all over the time. Well, I don't know what it was. Another thing that geneticists look at is the pattern of common substitutions. And if you look at the CCR5 molecule, which is what this is here, there's about 25 variants that we've discovered in Africa, Asia, and in Europe, in addition to delta 32. 90% of them alter the amino acids. This is the opposite from what you see in every other gene, where maybe 10% alter amino acids and 90% are what we call silent mutations that don't change it. The reason is, when you alter the amino acid, it's like taking the hammer to a chassis of a Jaguar. It's usually bad, so it gets eliminated quickly, with one major exception. Genes that defend themselves against infectious diseases, like HLA, have a high frequency of substitutions because of the constant impact of emerging infections on populations that drive up the frequency of good defensive alleles. This we see clearly, and it was well established in the HLA system 20 years ago. We also see it in CCR5. In fact, it's stronger in CCR5. This is a gold mine of selective advantage. Why? Well, summary. It's a knockout mutation that occurs in high frequencies in Europe, missing in native Africans and East Asians. European populations have been pretty high. 20,000 is a low number. It's probably higher than 50,000. So this mutation was born in some baby sometime in the last 40,000 years, and it had a frequency of 1 in 50,000. Today, 1 in 5 of you carries CCR5 delta 32. 1 in 5. How did it get from 1 in 50,000 to 1 in 5? Something caused it to go up. It wouldn't have gone up easily statistically. The statistical genetics, the possibility of that is very low. There was something good about having delta 32. What was it? Well, it probably was something like HIV, which killed everybody that got it, and the people who had delta 32 were protected. But it wasn't HIV, because HIV's only been around since, the early part of the 20th century in Africa, and maybe the middle of the 20th century in Europe and in America. Only a couple generations, not long enough to change the frequency. So, everybody that I talk to who study this agrees that delta 32 was elevated in recent history by natural selection. But what caused the selection event, and when was it? Another piece of data. Well, delta 32 is a mutation in a gene in CCR5. There's a whole bunch of other variants in CCR5 delta 32 that are listed right here. And we can measure how long the non-randomness is, because non-randomness is a function, is decayed quickly by recombination over time, unless there's a strong selective pressure, which all of a sudden sweeps up the frequency of something like delta 32. So we looked at delta 32 and all the variants around it. Said, how long is this chunk of non-random linkage disequilibrium? And the answer is it was about 1 million base pairs long. The average size of the chunks of LD in the human genome is 16 kilobases, 16,000, not 1 million. This is really long. So David Reich and Clay Stevens developed a simple little formula, which is on the top here, that you can run on your calculator that you buy from Walmart, that can allow you to calculate back how long, how many generations ago, or how many years ago it was since this mutation was born. Well, not since it was born, since it was last the subject of a strong selective pressure. So we did that. And the number came out to be 682 years ago. Now, I don't want you to write that number down, because there's an error associated with it. But it wasn't 6,000 years ago, and it wasn't 50 years ago. It was around 600 or 700 years ago. So I went to the library. What was going on in Europe 700 years ago? Well, something really big was going on in Europe 700 years ago, and that was called the Black Death. The bubonic plague at that time was marching across Europe from 1348 to 1352, and it decimated Europe. 1/3 of Europeans died in a 4-year period of time. Furthermore, that plague came back 10 years later and had almost equal mortality across Europe. It's said that 60% of the European population the population was reduced by 60% in the 200 years after the Black Death, until the Great Plague of Europe, which was in the 18th century. Furthermore, every 100 or so years before the Black Death, there were waves of plague back until 700 BC, when there was a 200-year period called the Justinian Plague. And that Justinian Plague killed up to 30,000 people in Constantinople every week. The number of deaths attributed to the Justinian Plague, I mean, I wasn't there. But the number that you go into the library, big library, and you'll see the whole, there'll be 6 feet of books about the Black Death. The number of deaths that have been estimated by Daniel Defoe and others was 100 million deaths caused by the Black Death. Well, 100 million deaths, I mean, I think they counted these with Roman numerals. It's kind of tough. But the point is, it was a lot. It was a lot of mortality, and it was in the right place. And we thought, maybe CCR5 led to some kind of protection. So now, I'm going to lighten it up a little bit. And I'm going to read you a couple of paragraphs from a chapter in this book. It's not about endangered species. It's about a woman who lives in Eyam, a hamlet in England in the 17th century. Her name was Margaret Blackwell. She was trembling and feverish and afraid. It was just before dawn, and she had suffered a fitful night, dozing, praying, but mostly crying. She prayed so hard that she developed a throbbing headache and was slipping towards hopelessness. Over the past few weeks, Margaret Blackwell had helplessly watched her son, her two daughters, her uncle, and her cousins wither and perish from the dreaded pestilence. She lived in the tiny English hamlet of Eyam in Derbyshire. Margaret was 36. The year was 1666. In the face of a growing plague, the 28-year-old vicar of the Eyam parish, William Mompesson, a devout and domineering prelate of the Church of England, had pronounced a cordon sanitaire, a strict quarantine for the 80 parish families to their homes in Eyam to limit the epidemic spread. Food provided by the Earl of Devonshire was delivered to a remote junction at the southern limits of the village, exchanged for the villagers' meager coinage soaked in vinegar. Mompesson announced that the church would be closed, services would be held outdoors. Burials and funerals were stopped, forcing the villagers to bury their loved ones in their fields and gardens. The terrified citizens agreed to isolate themselves for the good of others, as prescribed by the pastor. Now, the minister, he couldn't have known this, but his degrees were worse than useless. Fleas lurking in the fur of village cats and black rats readily spread the bubonic plague. The vermin would transport the disease from house to house as freely as the sun would shine through the windows. In a twinkle of historic time, every devout citizen of Eyam who remained became exposed to Yersinia pestis, the plague bacillus, the same deadly bacterium that had ravaged Europe 3 centuries earlier, causing the Black Death. Margaret Blackwell pulled herself from her bunk and hobbled down the loft ladder to warm her trembling body near the slow-burning hearth. She tried to move quietly so as to not disturb her mother and her brother, who were mercifully still sleeping. Now, Margaret dared not question the wisdom of the Creator, who has taken so many lives in such a horribly painful manner. She had heard of the Black Death ages earlier ago, and she had heard of the Black Death ages earlier, and of supplicants who would flagellate themselves in public piazzas of France and Italy to appease God's ire. She had prayed so earnestly her knees were bruised and inflamed. Or was the tissue inflammation part of the disease? She spotted the urn of bacon fat on the mantel, saved for cooking. It was still tepid and fluid. In her delirium, this is a true story, she thought the cooking fat would surely kill the demons that had possessed her frail and dying body. Maybe it would finish the evil within her, or at least shorten the agony. She drank the whole jug. Before she could contemplate her recklessness, she doubled over and began to vomit, to screech, to wail, and to whimper. Exhausted, she passed out, certain it was her last breath. But it was not. In a few days, her strength slowly returned, her fever subsided, and she rose to nurse her stricken family and neighbors. The death toll in the tiny village was staggering by spring. Of 80 households, 70 had at least one victim, most had more. The burials would reach 280 by the end of 1666, 50% of the populace of Eyam. My visit to Eyam in 2003 to the Eyam churchyard provided a grim image of those horrid years. Rows and rows of headstones bearing the years of death, 1665, 1666. These were the plague stones. 100 miles to the south in London, tens of thousands succumbed to the massive 17th century plague. In the spring of 1666, the Great Fire of London probably played a role in ending the dying. Yet not everyone who contracted the disease had perished. What had saved them? Was Margaret Blackwell really cured by the whopping dose of bacon fat she ingested? Blackwell's great-great-great-great-grandniece, Joan Plant, is today the administrative director of Eyam Parish Church. Joan, who told me this story, thought the radical potion might have saved Margaret. When we met, I thought it was CCR5 Delta 32. Here's why. The Black Death started when a ship came into Messina, Sicily, and unloaded carcasses, and black rats ran in. Then they went into Italy, and it spread quickly throughout the area until 4 years later, a quarter to a third of the population of Europe had perished. It was a horrendous, horrible death that affected history, politics, and put a grim face, really, on the entire population. It had been brought in from Asia, probably from ghettos in East Asia, brought in by black rats, and it originated in marmots. And there was basically well known as a disease that started out by infecting the lymph nodes and causing them to swell up to form the buboes, which is from which it got its name. And then it would spread throughout the body, causing fluids to come out of every orifice and causing a horrible death. The only solution that they had was to pray at that time. There was no other thing they could do. Everybody agrees that Delta 32 was selected by something. Not everybody agrees that it was the Black Death. I think it was because the timing was right, because the frequency was right, because the age of the mutation was right, and because the location took place in Europe, and because of a few other things I'm going to show you in a minute. But there were other conspiracy theories that came around about what was the cause of the Black Death, or what caused CCR5 to rise up. Was it smallpox, which also caused horrific death, or anything? There was a book in 2001 where some historians in England claimed they thought it was some sort of Ebola-like hemorrhagic fever virus. They weren't sure what it was, but they wrote a whole book about it, and they didn't have any data, they just said it. Couldn't argue with them because they passed away, both of them, soon thereafter. A few years later, Stephen Elvin and his colleagues published a paper in Nature where they basically, and I'm sorry for the color of this, but let me try to walk you through it. They wondered whether or not Yersinia would have an effect on individuals that had different genotypes for CCR5. They knew that mice were sensitive to CCR5, and they knew they could knock out CCR5 in a technology called knockout mice, which they did. And then they infected Yersinia into mice, and they measured the difference between the mortality of mice that had normal CCR5s and those that were knockouts. And they did the experiment several times, but as you can see, that the colony counts of the cells was 10 times higher among the individuals that were wild type versus negative for CCR5. This is a measure of the ability of Yersinia to infect mouse cells. CCR5 is required for Yersinia to get into these cells. This hasn't been done in humans. For technical reasons, it's been very difficult. I've tried, believe me. I work at Fort Detrick, which is a place where they work with spooky things like plague and things like that. But I wasn't able to get this done very well for technical reasons. But in mice, Yersinia requires CCR5. Anthrax. I dreamed of doing an experiment to see whether or not CCR5 had an effect on smallpox. But as you will recall, smallpox was eliminated entirely except for a stash in Russia and a stash at the CDC in Atlanta. And they were claiming that it was extinct, that it was cured. Then along came 9/11. And then people who were interested in bioterrorism said, well, I don't think we should throw this stuff out because we need to have the potential of defense against it. So they started doing experiments down at CDC. So one of my colleagues, I walked up to him and I said, well, why don't you do this experiment? Why don't you I'll give you cells from people who are heterozygous, homozygous for Delta 32 or homozygous for wild type. You see if there's any difference between smallpox infecting them. So down at CDC, they got permission, they did the experiment. The results are shown right here. Basically, as you can see, the three genotypes are indistinguishable. Smallpox doesn't care what the CCR5 genotype is. So I don't think that the argument about smallpox is holding much water. So, in sum, CCR5 delta 32 is a knockout mutation that seems to be innocuous to its carriers. Many of the people who come up to me and said they were homozygous for delta 32 look pretty healthy. Several of them lived here in their 70s. That's good. But they are resistant to HIV. It was born about 600 years ago when the population was probably close to 100,000. CCR5 showed dramatic evidence for breathtaking selective elevation. CCR5 is required for Yersinia and HIV to enter cells. CCR5 has no effect on smallpox. And I didn't tell you the results of this. We genotyped the people in EM. And the frequency of heterozygotes for CCR5 was twice as high in that population, which has been isolated, and there hasn't been a lot of immigration since the Black Death. And you can go back on the registry with these people that we measured and see what their relationship was to Margaret Blackwell and the people that were there. So they had an elevated frequency, even in EM, of delta 32 compared to the other people around there. So clearly, Yersinia seems to be, which is the cause of Black Death, the driving force that drove this thing up during the Black Death from the Justinian plague and forward. And people have dug up victims of the Black Death and discovered that they had, several of them were heterozygous for delta 32, but none of them were homozygous. In fact, there were graves that were a couple thousand years old that had delta 32 homozygotes, which means that the mutation was indeed born before the Black Death. But the last time it was really jacked up, selectively, was the Black Death or the Great Plague. Is it over? Not quite. One of my friends at NIH, Phil Murphy, was doing some experiments to see, you know, this CCR5 has got all these mutations that look like they've been selected. Maybe there are other diseases that also have co-opted CCR5. So he's going through a bunch of them, and one of them he looked at was West Nile virus. West Nile virus is mosquito-borne. It causes a rather nasty hemorrhagic fever. It arrived in New York City about 13 years ago. There have been 16,000 confirmed cases, of which 700 have been fatal. They progressed to encephalitis to 20%, heterogeneity, just like any epidemic. To date, there's no effective vaccine and no treatment, and it's carried by a mosquito. So what Phil did is he had some mice that were knocked out for CCR5, like the ones Stephen Alvin had. And he infected them, thinking, okay, if CCR5, delta 32, is protecting against West Nile, then the knockouts will be protected. That's not what he found. What he found is the knockouts all died, while the wild types were doing fine, the ones that had 2 copies. What does this mean? It means CCR5, a chemokine receptor. What's a chemokine receptor? Chemokines are short peptides that are secreted in regions of the body that get involved in inflammation. They're either infected or they're bruised or something. They're inflamed. You've all felt them. Sore muscles. And the lymphocyte brigade, which is scrolling by, has these receptors on the surface. And when they hit a chemokine receptor, they migrate to the site and deal with it. That's why it's been explained to me. So fine. There's a bunch of chemokine receptors. There's a bunch of chemokines in the human genome. CCR5 is one of them. If you knock out CCR5, you're pretty healthy. The reason is that the CCR5 specificity, which chemokines it binds to, is redundant. There are other chemokine receptors that do the same thing. But still, it's a good function. It's important. Well, this shows that in mice anyway, seems like CCR5 might be the most important thing for dealing with West Nile. So if you're a mouse and you get West Nile, you better hope you have a good CCR5 and not a knockout, because they all die. What about people? Well, it's a small study, but there has been some studies of the West Nile patients, and the frequency of homozygotes for delta 32 among the individuals that were negative for symptoms but were infected with West Nile was 1%, like everybody else. But the people that had symptoms, well, in Colorado it was 8%, and in Arizona it was 4%, or the odds ratio was 6 times higher overall. You're 6 times more likely to get hemorrhagic fever and encephalitis if you're Caucasian. If you're a mixed race, it's the same thing. Your risk for West Nile is elevated hugely. Most of the people who died of West Nile virus had delta 32. So if you're delta 32, you may be okay if you're exposed to HIV, but don't get bitten by a mosquito at a cocktail party, because this could do you in. It's a yin and yang thing. This is what we're understanding more and more. Finally, there's never been a cure for HIV. It's a disease for which we have no vaccines. We have treatments, but those treatments are designed in an evolutionary fashion where 3 drugs are added at once to specifically target the virus and keep it down. But when you suspend the treatment, because HIV infects maybe 30 reservoirs, 30 different tissues, it invariably comes roaring back. Enter Timothy Lee Brown, HIV patient, infected with AIDS in Berlin. Walks into his doctor, says, I got HIV. And they put him on the drugs. He didn't like the drugs. They didn't agree with him. He stopped the drugs. This was in 2001. Stopped the drugs. 2 years later, he comes in. He developed lymphoma. That's not a good thing to have if you're an AIDS patient. In fact, it's a death sentence. His doctor was a very courageous clinician named Gero Hutter. He says, I'll tell you what I think you need a bone marrow transplant. Bone marrow transplants sometimes succeed with lymphoma patients, not always. 1/3 of them turn to graft versus host reactions, and the graft kills the patient. But he had no choice. He was going to die anyway. So what Hutter did, Hutter screened the bone marrow banks. They have these big, huge blood banks in Europe and the United States where they type people for their HLA, and so that there'll be a match for a bone marrow transplant. And then if they get a match, they call a guy up, and he comes in, he donates the bone marrow. So they found about 200 people that were compatible with Mr. Brown. But Hutter wanted to do something more. He says, why don't we screen all those people and come up with somebody in that donor category who's also homozygous for delta 32 who will resist HIV, so at least the marrow will have a chance to get up before the HIV gets it. So he screened 100 people. Number 63 was a homozygote for delta 32 and compatible HLA-wise. Call the guy up, brings him in, says, would you be a bone marrow donor? He says, sure. He does the infusion, 2004. And he starts, of course, before that, he irradiates Mr. Brown to kill off all of his cancer cells, kill off his entire lymphoid system. Then he does the bone marrow transplant, infuses it into the guy, into Mr. Brown. But then he suspends the powerful antiretroviral treatment because he wants to give the bone marrow a chance to come up. But he monitors Mr. Brown for HIV in his blood so that he can start the treatment as soon as it comes back. So the first week, he tests him, he's negative. Tests him again the second week, he's still negative. 4th week, he's negative. 6 weeks, he's negative. 8 weeks, he's negative. He begins to think, it's interesting. Why isn't it coming back? The bone marrow transplant is doing well, the lymphoma's gone, it's recovering all right, Mr. Brown's feeling better. 12 weeks, still negative. A year later, still negative. A year and a half later, still negative. What happened? The graph was working. It had grown up. Guy shows up at the CROI meetings, this Conference for Retroviruses and Opportunistic Infection. It's the American version of the AIDS meeting. He gives a poster because they didn't think it was important enough for him to give a talk. I'm walking through the posters and I see this thing. And I says, wait a minute, this is 2.5 years now. This guy's been free. I says, did you cure this guy? And I said, I don't know, it looks like it. A couple months later, I get a call from Wall Street Journal reporter. What do you think about this? I says, I don't know what to think. I says, it certainly does defy my imagination. Then he publishes a paper. March, by now, 4 years, completely negative. They get together all the AIDS experts they can find, the best molecular biologists, people that can detect a half of a virus per milliliter. And they screen this guy. He's negative. They talk Mr. Hutter into allowing these guys to take biopsies in the weirdest places to see if any of his tissues, his kidneys, his testicles, his brain, his armpits, his lymph nodes. He just agrees to all this. Still all negative. They are now calling this the only cure of HIV that ever took place. It's not a cure or a proof that this works because it's just one guy. It's an anecdote. We don't know if something happened that made him clear it out, but it certainly was a proof of principle that Delta 32 as perhaps a target for gene therapy. So now there's several labs, Philadelphia and in California, who are doing the following. Before I tell you what they're doing, let me tell you the problem. They're not going to do a bone marrow transplant on a lot of people because of the GVH problem, the graft versus host. 1/3 of them die. This is not the kind of therapy that I'd like to talk you people into. However, it basically does mean that if you had an immunological match, then it might be. So what they've been doing is using the so-called zinc finger knockout technology, which allows you to go into the human cells of a blood, which you take from an AIDS patient, and knock out his CCR5, and then re-infuse him with this synthetic Delta 32. And those experiments began about 6 months ago, and so far, some of them are promising. I can't tell you where it's going to go, but it's the first time I've ever heard anybody talk about a cure for HIV, and it's based upon the discovery of CCR5. There's other promising things. This is an acronym for prophylactic exposure, pre-exposure prophylaxis. Robert Grant of San Francisco published a paper in New England Journal about a bunch of gay men that he had enlisted in a trial. It was real simple. They were uninfected. He gave them an antiretroviral cocktail called Truvada, which contained 2 powerful antiretrovirals, and then he followed them for 3 years. And what he discovered was after 3 years, the people who were taking the drugs had 48% reduction in transmission. But you know, part of these kinds of clinical trials are bad because they require compliance. And not all these guys, particularly if they're not infected, are going to be taking these drugs every day. But they were smart. They monitored the ones that were compliant. When they did the ones that were compliant, 95% reduction in HIV transmission. This is high-risk exposure, unprotected sex in gay men. 95% reduction. 2% had side effects, very mild. The cost was $150 a year, and the development or resistance against this thing is very, very low. We've never seen anything like that before, but it's promising. Pre-exposure prophylaxis also works in men who have sex with men. In this particular study, this is the study I just told you about, it also reduces infection among heterosexuals in Africa Two randomized studies found that daily antiretroviral therapy can reduce the risk. Treating uninfected individuals with one or two antiretroviral drugs reduces the risk of acquisition in heterosexual situations in Botswana and in Namibia and in South Africa by 70%. In addition, a number of studies have shown that circumcision is a way in which you can prevent transmission. The circumcision physically increases your resistance to HIV infection and transmission. So, and then there's been several antiretroviral vaginal salves that have shown to reduce HIV incidence and transmission in African trials as well. So things are better than they used to be. Okay, Tony Fauci said recently, in science, we have an unprecedented opportunity based on solid scientific data to control and ultimately end the AIDS pandemic. Major investments and implementation now save even greater expenditures in the future. In the meantime, countless lives can be saved. We now have hope. I've told you about Timothy Lee Brown's bone marrow transplant. We are working on vaccines. Some of them are promising. The pre-exposure prophylaxis is really working pretty well. Vaginal microbicides are working. In Montana, I can show this picture of President Bush, because if there is one legacy that I think he should be proud of and remembered for, it is for starting the President's Emergency Plan for AIDS Relief. In 2003, he got the Congress to give $15 billion for treatment and surveillance and monitor and capacity building in Africa. And in 2008, before he left office, $48 billion for the same thing. This has led to the treatment of several million, maybe 4 million people in Africa. It's a tremendous infusion of resources on a thing that works. Education has worked, and I told you the natural history story about it all. We're not at the end of this epidemic yet, not close, but we're closer than we have been in the generation that I have watched it. And now I'd like to thanks to some of the people who've helped me out with this work, including the organizations that have funded it, NCI, NID, Office of AIDS Research, the Laboratory of Genomic Diversity. On the right side of the slide are the cohorts, the epidemiologists who have spent a generation collecting patients, monitoring their clinical data, and allowing me to inspect the genetic associations. In addition to that, in our laboratory, these are the people that have run the GWAS, the Genome-Wide Association study, that have extracted the DNA, that have kept up the database and have moved it on forward, including the leaders, Cher Hendrickson, Jim Lautenberger, Bailey Kessing, Jennifer Troyer, George Nelson on Ping, Sherry Winkler, and Mary Carrington. And the analysis is now very, very computational. This is a list of many of the programs that we use, and these are things that we're starting workshops for in my new life, which will be involved in training some of the things that I've had the privilege of doing at NIH towards the rest of the world for gene discovery, for genome sequencing. And as Fred said, one of the things I'm looking forward to is a sort of a part-time commitment to the new Theodosius Dobzhansky Center for Genome Bioinformatics in St. Petersburg. And with that, I'd like to thank you very much for your attention, and I'll take any questions.
So there are 2 microphones up here. If you have questions for Steve, please walk up to the microphone.
Thank you. Could you give us, please, a quick review of the acronyms, the 2 positives, and how the Delta 32 relates to this? I got confused by that.
The acronyms.
You have the CCR5, plus, minus, and the Delta 32, and I got confused.
Oh, okay. When it says plus plus, it means you have 2 copies of the normal form. Plus delta means you have one copy of the mutant and one copy of the normal, and delta-delta means two copies of the deletion. Okay, those are the genotypes, sorry. Well, it must have been abundantly clear. Here comes a question, go ahead.
There was some research done on the CD4 receptors. Could you maybe talk about the direction that's going and if it's promising, or is that kind of a died-out route of inquiry?
CD4 is one of the receptors by which HIV enters cells. It's sort of like CD4 molecules on the surface of cells. They're part of the immune system that help the cells that bear them recognize viruses. And this is the cell that goes down in HIV-infected patients. HIV has to first bind to CD4, and then it sits there for a while, and then CCR5, which is this big, huge, meandering thing, kind of mumbles its way over and then binds to it, and that's what causes HIV to enter the cells. The search for inhibitors that interfere with HIV to bind CD4 have really not led to anything that looks clinically usable yet. And this is gets us into an area which a lot of people have a lot of opinions about, which is, you know, how much does it cost to develop a new drug, and what is the rate-limiting step, and why are some places better at it than others, and so forth. And it's just a can of worms which I'm thinking about a lot more and more these days, but I don't really want to get into it too much right now.
Over here, do you think that the receptors that are deleted for the cytokines are not engaging in what's called the cytokine storm, so that Yersinia causes this hyperinflammation, and the deletion of this receptor kind of reduces the inflammation? Is it something like that?
I think the cytokine storm illusion simply refers to you know, our lack of understanding as to what really is going on in these situations. I mean, when the chemokine receptor people first started doing their work, they were real excited about understanding that chemokines were excreted by areas of inflammation, which are, of course, fraught with cytokines. But when you try to get to what exactly these chemokine receptor-bearing lymphocytes do when they get to the areas of inflammation, it's not really clear to me. I don't think it's clear to them either. And so our understanding of the physiology of these immune phenomena is an area of very intense research, but it's not going as fast as I would like to see it go. So I don't think I can answer your question any better than saying it reflects more of a lack of understanding of process and physiology than I would like to admit.
Thank you for, it was very interesting. I was wondering, what, if you want to make an estimate, what percentage of heritability you think is in the susceptibility to AIDS progression, and what percentage of that heritability has been explained so far today?
That's a very good question. And it's a question that we have looked at very carefully, but I'd like to rephrase it. If you have a complex disease like HIV and you look at a particular step like AIDS progression, and you know that each of the genes that you discovered can be demonstrated and replicated to be real and causing an effect, what percent of the variation are you explaining? That is to say, how much of the disease can you actually explain by that gene? For example, if you have a disease like Tay-Sachs disease or sickle cell anemia, you can explain 100% by the mutation, because everybody who has the mutation has the disease, and everybody who has the disease has the mutation. But in these things, it's less than that. When you actually quantify the amount of variation that HLA and CCR5, delta 32, explains for progression, it's less than 1%. For infection, it's closer to 20%. That is, it explains 20%, which means that 80% we don't know why they're not getting infected. If you take all the genes that we know affect HIV, AIDS progression, and put them together and analyze their cumulative effect, as if the genotype itself could be quantified with the explained fraction, you still only get about 10 to 15%, which sounds awful. However, if you take cigarette smoking as a monitor for lung cancer, if you go into a lung cancer ward, most of the people are smokers. But if you go to all the smokers, not everybody gets lung cancer. And the explained fraction, using the same statistic, of smoking for lung cancer is about 10%. So just the genes that we've discovered explain about the same amount of the variation for AIDS progression as smoking explains for lung cancer. Well, that makes me feel a little better, but it doesn't make me feel good enough to say, okay, I can be predictive, because you really can't. The amount that we can explain that we don't explain is still 90% of the variation. Sorry.
Thanks a lot for that really stimulating talk. You said that in the CCR5 region there were quite a lot of not silent mutations, that is, mutations that actually had some effect on the actual amino acid
Yeah, they're
sequence.
amino acid altering mutations, right.
And that fraction was excessively high compared to sort of what you would expect at random.
Yeah, in general, the fraction of variance in a coding region of a protein, which actually alter the amino acid, is usually less than 25%. The only place that it's high is in genes that are selected by infectious disease that I know of.
Right, so is the thought that there are different diseases that have selected for each one of those 25 different non-synonymous mutations, or that it all got dragged up by the singular selective event of the Black Plague?
No, I don't think the Black Plague did it all at all, because many of them are found in Africa, which didn't have a Black Plague, and many of them are found in other places. I just think that the cumulative about the gene itself indicates that there's something about the fact that you can knock this thing out and be okay, which makes it a good target for viruses, because then they can hop on it and get in, and then the cell can knock it out by just making it redundant. So there's this evolutionary ratchet going on. This yin and yang is playing back and forth in an evolutionary dance, where you have the virus come in, and then the host adapts, and the virus adapts, and the host adapts, and we're just beginning to understand this dynamic by describing it. And I try to explain it in ways that a popular audience could understand. But there are technical details about these which are not always so easy to understand or explain. But I do think that we are now beginning in this genomics era to peel away some of these mysteries about why these genes and the genomes look as convoluted and as interesting as they do.
Thank you very much. In 1990, United Farm Workers, David Martinez described the suite of illnesses presenting in the farm workers in terms of pesticide AIDS. And then in 1997, Dr. Paul Repetto, in his book, Pesticides and the Immune System, referred to some Russian studies in which the researchers found and described AIDS-like illness in the absence of the AIDS virus. And you've spoken here in terms of unpredictability. I'd like you to comment, if you would, on what Repetto is saying and what Martinez is saying in terms of a healthy immune system.
Well, HIV is not the only thing that can cause a depression of the immune system. We know, for example, that cancer can do that, and cancer drugs can do it. So there's plenty of things in the absence of HIV that can cause the immune system to collapse. But when you have a healthy 30-year-old man who works out all the time and he's in perfect health, and all of a sudden he becomes susceptible to yeast infections or candidiasis or starts getting purple blotches or pneumocystis infections, you think, what's going on here? And that's really what was the beginning of the AIDS epidemic in the early 1980s. So that there are other epidemics of non-HIV associated immune collapse is something that we all know about, but that doesn't change our knowledge that HIV is not a virus that you want to become infected with, because in most people, but not all, the immune system collapses and you become at risk for some sort of a fatal infection or opportunistic infection or a cancer. And that's as simple as that. And if you stop the virus and you inhibit it with the drugs that we have right now, then in some cases the CD4 counts actually grow back, not all, but some. And it basically seems to be a treatment that has been more effective than ever. And with the President's program, the PEPFAR program, we are actually spreading this to the developing world at a cost of under $100 each, thanks to people like Bill Clinton and the motivators of the AIDS treatment.